All right, this is chapter 24 of ATI, and even though we're only going to cover, the chapter is cell wall antibiotics, and we're going to talk about some cell wall antibiotics, but there are only a few, so there are 4 of them here. But we need to cover some background, some terminology, stuff like that, so things make sense, and just in case you can't remember from microbiology class or whatever. So a pathogen. What is a pathogen? A pathogen is just simply something that causes a pathology. So it's an organism that can cause disease. Now, there are organisms, bacteria especially, all over the place, and fungi, but they're all over the place, but not all of them cause diseases. You have them living on your skin, they're on the table, they're everywhere. But if they can cause a disease, then we call it a pathogenic bacteria or a pathogen. So yeah, I mentioned bacteria because we're talking right now in the next several sections about antibiotics, but it can also include viruses, fungi, prions like Creutzfeldt-Jacobs disease, which is prions are just proteins that are kind of interfering with other proteins, and then multicellular animals, and we're not talking about polar bears or something. We're talking about nematodes or worms a lot of times. So those can be parasitic and they can kind of get in there and they're multicellular. So, but they can also cause pathologies or sickness. Disease. So cause disease in 2 different ways. Invasiveness, which means that they just divide rapidly and they overcome and cause direct damage. They steal resources, whatever, and that's how they can cause disease. Or they could produce toxins. I don't know if you remember this, but in pathophys we talk about endotoxins, endo meaning in. Endotoxins are just things that the bacteria produces and then releases, and then that can cause damage to cells or kill cells around it. There are also exotoxins, which are just part of the bacteria that make up maybe that peptidoglycan layer, but that can cause a inflammatory response or which can also lead to cell death. Okay, so endotoxins, exotoxins, those are just the production of toxins. So a couple of other terms, pathogenicity, okay, so that genesis, if you think genesis is the origin, pathogenicity is the ability of the organism to cause an infection, okay, so that's what that is. Virulence is more like potency, how potent it is. No, it's not a virus, but, I mean, viruses can be virulent, but really it's just the potency. So if it just takes a few little bacteria to cause a massive disease response, well, we would say that's very virulent. If it takes a whole bunch of them have to get in with no competition, then we would say it's not very virulent. You can overcome that if your immune system is strong enough. Okay, so if it's not very virulent, usually your immune system can fight it off. All right. Methods of describing, so I don't want any PTSD from your micro class when you look at this, but there are basic shapes, bacilli, cocci, spirilla, and these are just different shapes that they come in. You don't have to know all these. You don't have to think when you see, when you see it described as a bacillus type of, bacteria, you don't have to think, oh gosh, I need to know that looks like a capsule pill or something. You don't have to know that, but that's how they're categorized. And then we see the terms aerobic, anaerobic. Hopefully we understand this by now, but aerobic just means that it needs oxygen, and anaerobic means that it can survive and even thrive without oxygen. And then the type of cell wall. Hopefully we remember this from microbiology, gram-positive or gram-negative. If they have a really thick cell wall, then what happens is that there's a certain dye that gets in there, and that's the gram staining, and that dye gets trapped. Okay, so it gets kind of taken up by the organism. Gram-negative have a thinner cell wall. Okay, they have a thinner peptidoglycan layer. We'll come back and talk about that, which means that they have a harder time holding on to that dye. Okay, so that means they gram stain negatively. Okay, so that's the only difference. What it means for, I guess, the drugs that we're talking about is, well, if they're targeting the cell wall, they're probably going to be more effective against gram-positive types, although there are different formulations now that can make them also somewhat effective against gram-negative. Anti-infective drugs affect the target organism's structure, metabolism, or life cycle. So we read that and we say those are just words, and they are. Well, they aren't. So when we talk about structure, so there's maybe an E. coli, And it has, if we're talking about the structure of it, maybe we're probably talking about disrupting the cell wall. And if we disrupt the structure, the cell wall of it, then that organism can break. And then, because there's a lot of osmotic pressure inside bacteria, so if we weaken that, we weaken it structurally, then it can break. Metabolism, well, these guys have to also produce a bunch of nutrients so they can survive. You know, they make several gene products, and folic acid is one of those, and that folic acid is needed, just like in humans, for DNA division. And so without folic acid, then it can't survive. Okay? So we can interrupt that metabolism, that metabolic process of making folic acid. Life cycle. Well, they also have a life cycle. They divide and they replicate. They have the circular DNA, and if you don't let them divide that DNA, then again, you're killing it. Okay, so that's going to interrupt that life cycle. So these terms all make sense, and we're going to come back and refer to them. So antibiotics, what are they? Natural substances produced by other organisms. This is interesting. Bacteria kind of have a turf war going on, this bacteria, this bacteria, this bacteria, and then maybe a different type of bacteria moves in and says, okay, I'm going to produce something that kills all of you guys off so I can thrive. Well, guess what it did? It just gave us an antibiotic. It said it has the ability to kill other bacteria, so we can say, okay, well, we'll just use that, and we'll kill that. Or use that to kill bacteria, so we steal it. Even more effective are fungi. So remember with Alexander Fleming and his little petri dishes, he had a lawn of bacteria growing. So he had a bunch of bacteria growing, but then mold started to grow on it, and he noticed that the mold was killing the bacteria. Okay, so around the mold, the bacteria couldn't grow because the mold was making something. So we stole it. We took it. And that's what gave us penicillin. Just took it from the mold because the mold was making something that killed the bacteria. And so now we have antibiotics. Just that simple, isn't it? So a couple of terms when we're talking about things that either kill bacteria. Ultimately, that's what we want to do. We want the bacteria to die. But there are bactericidal, which makes sense. They actually will kill the bacteria. But then we have bacteriostatic. Static just means stationary. So that's going to slow the growth of bacteria. So normally bacteria divide at a pretty good clip, especially kind of early on. Well, what bacteriostatic agents do is they kind of slow that division, and then they keep it at a lower level. And then your immune system can kind of jump in and take it down. Okay, so that's the idea. Bactericidal, kill it. Bacteriostatic, just kind of slow the growth, stop the growth, so your immune system can kind of get an upper hand on it. All right, so let's talk about these mechanisms. I'm going to go through in the order that it says here, even though your book kind of goes through in a different order. Bacteria have DNA, and that DNA has to divide, which I know I just said I'm going in order, but I'm going to get this one out of the way. So the DNA has to divide. Well, there are drugs that just block that cell division. It blocks that replication, and that's number 4 here. But back to 1, the DNA is there to code for proteins, just like in every other organism. So the first thing it has to do is it has to make messenger RNA, okay, just like in people. It has to make the messenger RNA, and then the ribosomes bind to the messenger RNA, and then they start to make proteins. Okay, that's what we should have learned in physiology class. So there are drugs that don't allow this RNA to be made. Rifampin is a drug, RNA synthesis inhibitor. Protein synthesis inhibitor, well, what are we doing with the mRNA? The purpose of the mRNA is to make proteins that the bacteria can use. So what if we blocked the ribosomes and we said, no ribosomes, you can't bind to the mRNA, or no ribosomes, you can't move along that mRNA? And if that's the case, then you're not going to have any proteins. So that would be a protein synthesis inhibitor. Antimetabolites, well, what are these proteins? Some of these proteins are enzymes to make things like folic acid. Okay, so if you can't make the enzymes, or you block the enzymes that make the folic acid, Fa, so if you block that process, then there's no folic acid made, which means you can't have DNA replication, which we already talked about. And then One of the other things that's very, very important specifically to a bacteria, which is why it's such a cool drug, is a cell wall. People don't have cell walls. Eukaryotic cells don't have cell walls, which is what we have. We have eukaryotic cells. We don't have cell walls. So all we have to do is block the process of making cell walls, and hey, it's kind of like magic. We're killing a process that we don't even do, so we don't care. But the bacteria care, so it breaks down those bacteria. All right, which is what we're going to talk about is cell walls. So resistance, we can't not talk about antibiotic resistance. Mutations happen. Okay, so mutations in these bacteria happen. And so even though the bacteria start out as one form doing things one way, if you start taking antibiotics, okay, now chances are your immune system will kill the resistant organism too normally, because it happens all the time. But if you have an infection and you're kind of overrun and your immune system isn't able to keep up, then we can take antibiotics, and those antibiotics will target the cells that are susceptible to the antibiotic. But sitting over here is a resistant one. Now, the idea is for, taking antibiotics is that you have to take the full dose and you have to take it until it's complete. You have to really kill them off, because what happens is if you just kill a few of these, okay, and the patient starts to feel a little bit better, well then what if your immune system is still going to be like working, even though the patient may feel better, their immune system is still kind of trying to get this under control, and there's a resistant one here. And so that resistant one has the opportunity to start dividing, and then it can take over, and then you end up with, so if you're this guy here, you're in trouble. This is a petri dish. But if the resistant form is able to take over, then you're going to, giving the same antibiotic again isn't going to have any effect. And so you're going to have, because now it's resistant. Okay, so antibiotic resistance occurs when a pathogen develops a gene and survives longer or grows faster. Okay, so something gave it an advantage to survive in that environment. So through mutation, antibiotics destroy sensitive bacteria, but they're destroying the sensitive ones, but the insensitive or the mutated, the resistant one stays. And then it doesn't have as much competition. The immune system isn't seeing it, or maybe doesn't, hopefully for it. And then it goes on. So random mutations occur during cell division, may harm or help bacteria. Mutated bacteria multiply, and then they may pass on that resistance. They can actually kind of, the bacteria around it can kind of take up that resistant gene and integrate it into their own genome with that reverse transcriptase, remember? And when it does that, then others that weren't resistant to begin with can become resistant. So yeah, it's a real nuisance. They're crafty little guys. So widespread use of antibiotics contributes to resistance. Remember, the antibiotics don't create the mutations. Mutations are just happening. The antibiotics are just selecting out. They're killing all the susceptible ones, and they're letting the resistant ones thrive. So resistance isn't caused by, but it's worsened by overprescription of antibiotics, results in a loss of antibiotic effectiveness. So the idea is you only prescribe when necessary. You don't give it for things like viruses. A virus, it's not a bacteria, so antibiotics won't work on it. But there was a time, and that's getting better, but there was a time where somebody would come in with a little sniffle, and the physician, nurse practitioner, whatever, would prescribe an antibiotic and just say, there you go, now you didn't waste your trip in. Well, they did anyway. In fact, it was more harmful if they had just not, if the prescriber had not given them that. So long-term use can increase resistant strains as well. Hospitals are a breeding ground a lot of times for resistant strains, so that's something to be cautious about. That's why we try to keep them sterile as possible. So healthcare-associated infections are often resistant. Prophylactic use is appropriate in some cases, so that means taking a bacteria before you go to pick your kid up from daycare because you don't want to catch anything is probably a bad idea. But there are certain times, dental procedures to prevent growth, vegetated growth on heart valves. That's something, now that would be a good prophylactic use of it. Okay, nurse should instruct the patient to take the full dose to completion. So that's kind of the take-home message there. So selection, it's important to select the correct antibiotic. Now, you can give a broad spectrum antibiotic, and a lot of times that happens if a patient is very sick, but especially in the cases of like pneumonia. Pneumonia have different organisms. Different organisms can cause pneumonia in the lungs. So if it's one type but not another type, then your treatment isn't as effective. So it's good to get this culture and sensitivity testing. So really, I'm not going to read through all this, but that's just trying to identify exactly what the organism is, so then you can find the right thing to actually kill it. Okay. So provide effective pharmacotherapy and limit adverse effects as well. All right, so selection of an antibiotic, broad spectrum, effective for a wide variety of antibiotics, and then there are narrow spectrum that are effective for a narrow group of antibiotics. And yeah, a lot of times they can be more effective to use that narrow spectrum because you're targeting the actual pathogenic bacteria, and you're not, increasing things like C. diff, which is kind of a supra infection. So, so that's kind of what we're at here. So secondary infections, those occur when too many host flora are killed by an antibiotic. So here you may see, a whole bunch of bacteria growing in the intestine, and with those, there might be a Clostridium difficile, a couple of C. diff there that can't grow. Okay. Well, then if you kill off all of its little friends, then that C. diff, which is pathogenic, but it's still there, it's just not causing any problems because it's being outcompeted, suddenly that starts to replicate because you've used an antibiotic and you've killed the competitors. Okay, so that's a supra-infection. That's a secondary infection. So host flora, so that means the little blue ones here in this case, normally prevent the growth of pathogenic organisms, but if pathogenic organisms are given a chance to multiply, so they're opportunistic, they take advantage of the suppressed immune system, something. Signs and symptoms include diarrhea, bladder pain, painful urination, or abnormal vaginal charge. All right, so let's start talking. Well, we're almost going to start talking about the actual drugs. So these are the classes that we're going to go through. Organizing the chemical classes: penicillin, cephalosporins, carbopenems. Those are the three, because right now, three classes we're going to talk about, because right now we're interested in the cell wall, and then we'll come back to those. Okay, one more sciencey thing to go over, and this one kind of matters because you're going to see this term here a lot: beta-lactam. It's a beta-lactam antibiotic. Well, it's kind of nice to know what beta-lactam is. It's this little structure right here, and penicillins have this structure, cephalosporins have this structure, so they're all considered beta-lactams. And what this does, what the molecule, what the penicillin does, is that, remember that cell wall? That cell wall has to produce these things. Now, I know this looks like a complicated picture, and it kind of is, but we don't have to get into it too much. But it produces these things called peptidoglycans. And these peptidoglycans all come together to make the cell wall. Okay? So what these beta-lactams do is they don't let that happen. So they prevent that cell wall from being made. They weaken the binding between these beta-lactams, and that's gonna kill the bacteria. Now we can see here, this is a better picture than I drew earlier. This is a gram-positive. See how thick the cell wall is? And this is a gram-negative, which barely has any of this peptidoglycan layer. And that's what this shows as well, the blue down here, the peptidoglycan layer. So really, a lot of these drugs were initially just they only worked on gram-positive, but they've changed the formulations a little bit, so then they now have an effect on gram-negative as well. Okay, so originally most effective against gram-positive, but different formulations include gram-negative. Resistance occurs, and this is what also you're going to see. Resistance occurs when bacteria produce beta-lactamase. Okay, so the bacteria become resistant because they say, oh, look what's killing us. It's this thing. So look what we're going to do. We're going to kill that. We're going to break that down. Okay, so that's what we're at war with. And so now this is a resistant form because it produces this enzyme that breaks down the thing, the molecule, that's trying to kill the cell wall. So we just take it out. Take out that beta-lactam with beta-lactamase, an enzyme. Breaks it down. So we're fighting back, and so we come up with this drug, clevulinate. Clevulinate blocks beta-lactamase. Okay, so do you see what's going on here? It's a back and forth. It's oh, we're going to kill you, bacteria. The bacteria says, no, we're going to make something that blocks your beta-lactam. And we say, no, we're going to block the thing that blocks our beta-lactam. Okay, so that's the battle that's going on. Okay, so when you see that, just know that. All right, so let's talk about these. We have 4 to cover. The first one are the penicillins, and yes, they're part of this cell wall synthesis inhibitor, part of one of the groups that we're going to cover. So penicillin G is the one. Penicillin, there's another formulation, penicillin V, that survives in the oral cavity, so that's used a lot for dental types of procedures. Mechanism of action, it's a beta-lactam that kills bacteria by disrupting that cell wall, and it blocks that cross-linking of those peptidoglycans that are surrounding it, that kind of make up the cell wall. So it binds to cell walls, which allows water to enter, just messes them up, messes up their ability to hold in all that water pressure. So humans don't have cell walls, so that's nice. Most effective on gram-positive, but they also, like I said, can work on gram-negative now too. Primary therapeutic use, gram-positive and gram-negative infections, meningitis, syphilis, prophylaxis against bacterial endocarditis. That goes back to especially dental work. And that's how they figured out a lot of people were getting this vegetation on heart valves. And this endocarditis is that it was entering through the mouth during dental procedures. And so, yeah, a lot of times they'll give penicillins prophylactically to prevent that. So really, they're the safest class of antibiotics, but they do have allergies, and allergies are huge for penicillin. Okay? Probably because they've kind of been in our ecosystem for a long time, and so we've learned to fight them off for whatever reason. Maybe we don't like fungus because that's where they're made from. But anyway, yeah, allergies are a big problem. It can also cause some renal impairment, hyperkalemia, dysrhythmias, hypernatremia with some formulations. So these things can happen, and may decrease the effectiveness of oral contraceptives. I didn't see that listed in your ATI book, but it's listed in other places, so I left it in there. Okay, so those are the penicillins. Cephalosporins, so cephalexin. Cephalosporins, let's see, yep, they're included here too. Mechanism, they are also beta-lactam antibiotics that destroy bacterial cell walls. Now, in your ATI book, you'll see different formulations. Okay, so different formulations, they're generations 1 through 4. They may have different routes of administration. Okay, so whether it's oral IM, IV, reach different targets. Like some of them have been modified so they can get into cerebrospinal fluid, which would be good against something like meningitis. Okay, which is listed down here. But not all of them can get into cerebrospinal fluid. So they affect different types of bacteria. Some affect gram-negative, better against gram-negative, better against anaerobic bacteria, and they're better against resistance. So this is kind of that constant competition to try to stay ahead of the resistance. And yeah, so that's what's going on. So adverse effects, allergies, cross-sensitivities with other penicillins, they're similar. Certain formulations, these aren't our prototypes, but may increase bleeding tendencies, thrombophlebitis with IV fusion, pain with IM injection, antibiotic-associated pseudomembranous colitis, inflammation due to overgrowth of C. diff. Okay, so that's what we're talking about. We're talking about inflammation that occurs in the intestines, in the colon, and that's associated with C. diff. So that would be a superinfection. Secondary infection. All right, carbapenems, imipenem, cilistatin. This is a combination drug, but essentially it's the same mechanism. It's a beta-lactam antibiotic that destroys the bacterial cell wall. So, but it's broad spectrum, gram positive, gram negative, used for serious infections such as pneumonia, peritonitis, UTI due to gram positive, gram negative, and aerobic bacteria. So resistance develops from using imipenem alone to treat Pseudomonas aeruginosus infections. All right, and so that pathogen requires that combination. So adverse effects, again, allergies, cross-sensitivities with penicillins and cephalosporins, GI upset, superinfection. So other inhibitors of cell wall synthesis, these guys work a little bit differently. They're not really a beta-lactam. And this is vancomycin. Vancomycin used to be the go-to drug, the last line of defense against MRSA. Okay, well, that's not the case anymore. Now there are vancomycin resistant strains, but they are included in this cell wall disruptor category. So primary uses, most effective against gram-positive infections such as staph, which is MRSA. Well, it's not MRSA. Staphylococcus that's not resistant is just Staphylococcus. It's this methicillin-resistant Staphylococcus that's the problem. So treat serious infections such as MRSA, methicillin-resistant Staphylococcus aureus. Treat antibody-associated pseudomembranosus colitis due to C. diff. Okay. So there's your treatment for C. diff. So you take an antibiotic, C. diff gets a foothold, and then you give an antibiotic to get rid of the Clostridium. Okay, fun. All right, so adverse effects. Autotoxicity is listed. It's rare, but it's reversible. Infusion reactions. So something called red man syndrome, which are rashes. It's a rashes all over, tachycardia, hypotension, so low blood pressure. So we're talking, so we're talking low blood pressure, and then the heart is trying to beat faster to kind of make up for that. And then the superinfection can occur with it. Treating it can occur with it. It's a mess. All right, so that's it for the cell wall bacteria and your whole background in microbiology.